In this session of Diagram Dazzles, we'll be learning about the monocot root in section. So here is a briefing. It basically has three regions: epidermis, cortex and the stele. Epidermis is outermost, characterized by the presence of root hairs. The cortex is wide parenchymatous and has endodermis, which possesses the Casparian strips. From the pericycle to the pith, the entire region corresponds to the stele. It has vascular bundles that is said to be radial. Xylem and phloem alternate each other in occurring in separate radius. Then the xylem is polyarch. As you can see, their number is always greater than 5. The pith is large and parenchymatous. So here we start with the tricks to draw the monocot root in section. So here is the outline. So draw a border line and then a circle wherein you demarcate the xylem by means of lines and then the phloem by means of semicircle. Now the association which could be developed for this is a simple one, that is the dial of a watch. So here the xylem corresponds to the demarcations of the hour and then the phloem corresponds to the minutes in between. So after you do the detailing, you can see the various regions: the epidermis, cortex and then there is the stele. So let us start. To begin with, you will have to draw an outline, draw an arc and then a circle. This circle should be quite large in comparison to what you draw for the dicot. And then set the regions for the xylem. Remember, it is polyarch, so the minimum number you need to draw is 7. And alternating, that is the phloem. So now we'll develop on this particular outline. First, draw the epidermis, small barrel-shaped cells across the border which you've already drawn, and then complete it. Characterized by the presence of unicellular hairs, show some outgrowths in some cells, and this is nothing but the root hair. After that, proceed with the drawing of the endodermis. So draw the endodermis like the epidermis, barrel-shaped cells, and this is the circle which is drawn. Next, you'll have to draw the pericycle with the gentle pressure of the pencil. As you can see, it should be drawn as light, thin cells. Complete this circle and then we go ahead with the drawing of the vascular bundles. Start with the xylem. At those points where you have already demarcated, draw the xylem. The large xylem corresponds to the metaxylem. Draw them elliptical and then after that complete it for all the 7 of them, and then you are now going to draw the inner lining of the xylem which has to run thick parallel to the outer lining. So this corresponds to the lignification which is found in the xylem elements. So once you've drawn that, next draw the protoxylem, similarly circular, very small as such, with the outer lining and then the inner lining, that is a secondary wall made up of lignin. So complete likewise for all the xylem bundles. Next we shall go ahead with the important lining for the endodermis. So this has to be drawn only after the protoxylem is drawn, because here you should be careful. So when you draw the Casparian thickenings, draw it for all the cells except those passage cells which are shown across the arrows, and you can see that they are positioned right opposite to the protoxylem, the small circular structures of xylem. So there should be the passage cell. That region has to be passage cell. Phloem has to be drawn polygonal, small cells like this, and then it should be dotted for some of the cells so that you can show the sieve pores across. So complete it for all the phloem bundles which alternate the xylem. So once that is done, the vascular bundles are all ready. Next, go to the pith. The pith is parenchymatous. Draw oval or elliptical structures, and this is parenchyma, extend across the center. So this is the parenchymatous pith. And last but not the least is the conjunctive tissue. Draw it polygonal, small, with a gentle touch of the pencil, and then fill in this region between the xylem and the phloem. Once it is completely filled, this is how it looks. Next, now go back to the cortex, that is also parenchymatous. Fill it in the similar manner. So once the filling is done, start with the labeling. The outermost is epidermis, it has root hairs, followed by the cortex. Show the endodermis. Importantly, not to miss the Casparian strips and the passage cells, and after that the various components of the stele: pericycle, phloem, protoxylem, metaxylem, conjunctive tissue, and the correspondingly form the stele.